All right, people. So, welcome to Jumbo IAS and welcome to UPSC Prelims Phoenix batch. In this batch, we revise the entire syllabus for UPSC Prelims examination that includes both static as well as current affairs. Now, this lecture is related to current affairs portion. As you all know, that we have started with science and technology current affairs. All right. And today is day one for science and technology. And your day one's targets include all the important current affairs related to biotechnology. Okay, so today's 20 MCQs would be related to biotechnology and the current affairs associated with it. All right, so let's revise those topics which you have already revised. Let's go for revision number 2 through a set of 20 MCQs. So let's begin. Okay, so science and technology current affairs day 1, and today's targets were biotechnology. Okay, so let's begin with question number 1. All right. With reference to regulatory framework of genetically modified crops in India, consider the following statements. Okay. The commercial release of GM crops is governed by Rules 1989. That's true. Enacted under Biological Diversity Act 2002. Genetic Engineering Appraisal Committee is the apex statutory body for allowing commercial release. Review Committee on Genetic Manipulation under Ministry of Environment monitors small-scale field trials. Okay. So here's the thing. Well, you can answer this question through logic also. Now listen to me very carefully. The first statement is absolutely false and you can easily understand why. The rules are Rules 1989. How can Rules 1989 be framed under an act that was enacted in 2002? That's kind of impossible, right? And it is. These Rules 1989 do definitely govern GM crops, but they were not framed under Biological Diversity Act. They were framed under Environment Protection Act 1986. Okay, so first statement is false because of that. Now, Genetic Engineering Appraisal Committee is the apex statutory body which is responsible for allowing commercial release of GM crops. That is true. And this Genetic Engineering Appraisal Committee, GEAC, functions under Ministry of Environment, Forest and Climate Change. Okay. Now, Review Committee on Genetic Manipulation, which approves small-scale field trials, is not under Ministry of Environment. It's actually under Department of Biotechnology. And Department of Biotechnology is under Ministry of Science and Technology. So this is also false. So only second statement is correct. The answer would come out to be A. Right? Next. In context of precision breeding in India, how do SDN1, SDN2 categories of gene-edited plants differ from traditional GM crops? Now listen to me very carefully. You see, GM crops, genetically modified crops, or GE crops, genetically edited crops, okay, they are created through gene editing. Now, first of all, you need to be aware of the difference. You need to be aware of the difference between GM and GE. Well, GM crops are those crops which are created by introducing a foreign gene into the plant, okay, from another organism, maybe a bacteria or anything. GE crops, well, no foreign gene is introduced. Okay, well, some gene is removed or added. GE crops, no foreign gene is introduced. Clear? Now, both these crops are created through genetic editing, gene editing. Now, there are various techniques for gene editing. Okay, two of these techniques are SDN1, SDN2. Now, SDN stands for site-directed nuclease. This is just a technique. You don't need to know what type of technique is it, how does it work. No need to know that. Just remember SDN1 and SDN2 are techniques. Okay, and these techniques are permissible. You don't need to take any approval from GEAC for these gene editing techniques. Okay, so they are exempt from stringent GEAC clearance process required for GM crops. No GM crop can be released without GEAC clearance. But GE crops created through SDN1, SDN2 techniques require no approval. Okay, so this is the difference. Answer is B. Okay, SDN1, SDN2 is genetic editing used for GE crops. So in GE crops, no foreign gene is introduced, and because no foreign gene is introduced, it doesn't require any approval from GEAC. Okay, so the answer would be B. Next. Regarding the status of specific GM crops in India as of April 2026, which of the following are correct? BT brinjal is the only GM crop under commercial cultivation since 2002. GM mustard, DMH11, Dhara mustard hybrid, has received final clearance and is currently being grown nationwide. BT cotton uses Barney's Barstar system to resist pink bollworm. GM mustard utilizes a hybridization technology that critics fear is backdoor entry for herbicide tolerant crops. Okay, so remember, no GM crop is allowed in India for commercial cultivation except BT cotton. And BT cotton has been allowed since 2002. So first statement is false. BT brinjal is not allowed. Now BT cotton is created by introducing two foreign genes. Okay, two foreign genes, CRY1AC, CRY2AB. These are the two genes which are bacterial genes. They are taken from bacteria BT, Bacillus thuringiensis. Hence it is called BT cotton. Clear? So, well, this is the case. Now, GM mustard has received final clearance and is currently being grown nationwide. No. GM mustard was created, but it has not been cleared and it is not being grown. Now, BT cotton uses CRY1AC, CRY2B genes, not Barney's Barstar. Barney's and Barstar genes are in GM mustard, DMH11, Dhara mustard hybrid. So this is also false because Barney's Barstar are for GM mustard and they make GM mustard herbicide tolerant. And yes, some critics argue that this gives backdoor entry. Okay, I mean critics who don't want it, don't want GM mustard to have clearance. People who are opposed to GM crops. But yes, it uses hybridization technology and using Barney's Barstar, it makes it herbicide tolerant. This part is correct. The only correct answer is D. Okay, next. Consider the following statements regarding Barney's Barstar system. It is a technology specifically associated with development of GM mustard. It aims to increase crop yields by approximately 25 to 30% through hybridization. It is a gene editing tool that falls under SDN1 category for precision breeding. Now remember, GM mustard is GM, which means it requires foreign genes. It uses foreign genes. And if it uses foreign genes, it cannot be either SDN1 or SDN2 because SDN1, SDN2, they are GE, not foreign genes. So this is absolutely false. Okay, but yes, this is correct. Barney's Barstar is GM mustard. We have already done this, and this makes the crop herbicide tolerant, which improves the yield by 20 to 30%. This is also correct. So the correct answer would come out to be A. Right? Next. With reference to Genome India project, consider the following statements. Phase 1 involved a whole genome sequencing of 10,000 individuals across nearly 100 ethnic groups. Phase 2 focuses on disease-based sequencing to compare healthy reference genomes with those of patients. All genomic data is stored at Indian Biological Data Center located in Bengaluru. Now, what is Genome India project? So, Genome India project is basically sequencing all the genes in Indians. It is for Indians. Now, India has many ethnic groups. Therefore, Genome India project would require genome sequencing. What is, list of every gene and its function. This is whole genome sequencing. Okay. So, you will be surveying around like 10,000 individuals across 100 ethnic groups because different ethnic groups have different genes also. So, this is correct. This is phase 1. Then phase 2 is disease focused. You see, some people are more prone to some kinds of infections like malaria, and some people are naturally immune to malaria. Not completely immune, but they are less vulnerable to malaria. Surely there is some mutations in their genes, right? So therefore, in the second phase, we'll compare the genes of people who are more vulnerable to particular disease and people who are less vulnerable to that disease. Okay, this is 2. And the entire genomic data would be stored at Indian Biological Center, which is not in Bengaluru, it is in Faridabad, Haryana. Okay, so this is false because of the location. So the answer would come out to be B, 1 and 2. Okay, next. The founder effect mentioned in the context of India's genetic diversity refers to. Now, what is a founder's effect? Okay, now look, India practices a kind of endogamy. Endogamy means marrying within community. So in India, intercaste marriages are not that common. Okay, I mean, traditionally people have been marrying in their own caste. Now, this is an example of endogamy within your community. Now, if there is a genetic mutation in one person, well, he will pass around that genetic mutation within that community because he is marrying in that community. Okay, so a person has some genetic mutation, well, that would be passed on to his son. That son would pass on to other people. Within that community, that genetic mutation, that genetic variation gets amplified. And hence, some caste groups have some different traits. Okay, because that genetic mutation makes them different and it becomes more common in that particular caste. It gets amplified in that caste. So amplification of specific genetic mutations within communities due to endogamy is founder effect. It happens. That is why it is generally prescribed that endogamy shouldn't be practiced. You should marry outside your community, produce offspring outside your community so that your genetic pool gets diversified. It's better. Okay. Nonetheless, this is B. Next. Which of the following best describes the difference between Genome India project and Phenome India? Now, here's the thing. For that, you need to understand the difference between genome and phenome. So, genome is genes, your genes. And because of your genes, you have different physical traits. Okay. So, if you have a particular set of genes, you'll be taller, you'll be darker skinned, you'll be thin, you'll be less susceptible to certain diseases. It's all because of your genes. So, your physical traits come because of your genes, are there because of your genes. Now, genes are genes. These physical traits, biological traits are called phenotypes or phenome. Okay. Now, Genome India project would sequence your genes. Phenome India would sequence the physical traits related to that. Okay. So, GIP focuses on static genetic code while Phenome India tracks longitudinal changes in clinical traits or clinical or physiological traits, physical traits. So, the answer is A. Okay. Next.